What's going on, y'all? Welcome back. It's another Whiteboard Wednesday. I'm super pumped for today because I finally got the courage to talk about something that I've been wanting to for a long time. This complex, really daunting disease known as systemic lupus erythematosus, or in other words, lupus. But May is Lupus Awareness Month, so I had to step up and do it. So we're talking about actually peering into the dysfunctional immune system. There's a lot into this. I'm definitely only going to be scratching the surface, barely, if even. And we're going to be talking about the labs associated with it, the genetic markers, triggers, and deep into the pathogenesis, or how it develops within the immune system. Truthfully, in my opinion, I think lupus is the most devastating and most difficult autoimmune disease to deal with. I've seen what it can do to people, and the fact of the matter is it can cause renal failure, kidney damage, and kidney loss, heart attacks, fatal infections, and premature mortality. The incidence of which is 3 to 13 times higher among women compared to men. And 90% of people who have lupus are indeed females. Now what's interesting is that African Americans have a 3 to 4 times increased risk of having lupus compared to Caucasians. Now, why is there this racial disparity? It's not really clear, but there are a variety of susceptible genes that have been identified, and it's probably what is underlying this great divide here. But none of the genes have actually been identified, so we can't really elucidate on that fully and explain why people with African descent have a higher chance of lupus. So looking at some of the clinically significant biomarkers for lupus, whether it's making a diagnosis or checking progress, we have anti-double-stranded DNA. This is a big target for lupus, and you'll see why in a bit. We have anti-phospholipid antibodies. These are antibodies against lipids found in your cells, virtually any cell in your body. And approximately 40%-ish of people with lupus have these. People have what's known as anti-phospholipid syndrome. Not everybody with lupus, but anti-phospholipid syndrome is a condition that is quite prevalent, actually. And it's autoimmune, of course, but what's happening is that there's an issue with making an overabundance of blood clotting proteins, essentially. So that's an issue because that can put you at risk for a stroke, myocardial infarction, heart attack, and basically thrombotic events. These antibodies can also attack what's known as a trophoblast, which is a cell that's formed during the beginning stages of pregnancy, so you get a high risk of pregnancy loss. Anti-nuclear antibodies, ANA, that's a popular one people talk about. Here's the deal with this one. A lot of people get really obsessed with this, thinking that they have a positive biomarker for this, they have lupus. 95% of people with lupus have this antibody. Okay, we know that. But the presence of ANA antibodies does not guarantee that you have lupus. All right, so let me say that again. It's part of the criteria, but it's not specific. It may be found in people who are just healthy individuals, okay? So you have to take it with a grain of salt and look at it in the bigger context of everything else and symptoms, of course. We have anti-Smith and anti-RNP. Smith is referring to proteins inside the nucleus that antibodies attack, and then RNP is referring to complexes inside the nucleus that makes proteins. And we have anti-Rho and anti-LA. These are, again, two complexes inside the nucleus and cytoplasm of a cell that contain RNA, and they're kind of coupled with proteins known as Rho and LA. Anti-C1q. So C1q is a component of the complement system that's inside of the overall immune system. So when you have an antibody-antigen complex, that happens all the time in autoimmune disease, that complex can bind and hatch onto C1q, especially in the kidney. And so we actually know that there's a strong association with this antibody and developing nephritis or kidney inflammation overall. HSCRP, C-reactive protein, it's a non-specific inflammatory marker. You've probably heard about this before. It's elevated in many inflammatory conditions, but actually in lupus, surprisingly, it can be low because there are cytokines that can influence the expression of this acute phase response. Erythrocyte sedimentation rate, usually high and implies inflammation, but it's non-specific like CRP. Complement, C3 and C4, we test these in Lyme disease sometimes, and the thing with this is that it can be effective and helpful, but it's a bit debatable. Because what can also happen is that they can get used up when they're fighting a lot of antigens, and levels can actually be low compared to high, like we might think. A urinalysis would be useful, and then a complete blood cell count, typically run by your doctor, because what we can check for is your hematocrit and your red blood cell and see if you're deficient in iron or there's some issue or anemic tendencies there. And then that would also check for your platelet counts and your white blood cell count, which are commonly low in lupus. As a culture, we have a tendency to assume that if we see these symptoms listed under a specific disease or condition, and we have them, we therefore have this condition. So there's an issue with that, okay? And the reality is that these symptoms I'm about to tell you are capable of overlapping with other conditions. So imagine you have swollen lymph nodes, hair loss, swollen glands, fatigue. That can look like mold toxicity, it can look like Lyme disease, Hashimoto's, and lupus. Right? But also fever, ulcers, edema, swelling, Raynaud's, poor circulation to your fingers and toes, and photosensitivity, right? Light bothers you. These are all typical signs of lupus. But again, you gotta look at it in the entire context of the person. The diagnostic criteria for lupus involves having a positive finding of 4 out of 8 of these things that I'm about to share with you. So we have to have abnormal cells in the urine, we have to have some sort of arthritis, arthritic issues, a butterfly rash, which isn't even that common, actually, right? White blood cell count has to be low, mouth sores, seizures perhaps, and then photosensitivity, like I mentioned before, and then specific antibodies. So a quick word here on genetics. I'm not going to go too much into this because it would be entirely too much, but what's important to understand is that there are 93 different genes roughly that we found to be associated with lupus. So there are some sort of genetic polymorphisms or variants that increase this risk. The most salient point I'll make is that the genetic variants associated with the major histocompatibility complex genes still confer the strongest genetic risk for lupus in the general population today. And you'll see more about that MHC thing later. I've talked about this extensively in the Hashimoto's development Whiteboard Wednesday, by the way. One of the ones that we can pay heed to is one known as HLA-DR3, and this kind of relates to MHC. And we know this is strongly linked to lupus, Sjogren's, and even myositis. I have a client now who has lupus and myositis and Hashimoto's. And we haven't checked for this, but she probably is positive for it. The other thing is that hypomethylation, right? When you're an undermethylator, think about MTHFR, that's something we've all heard about, right? People have a genetic snip of that gene, a lot of us do. However, hypomethylation is a common problem, especially in lupus, because people who are undermethylating, basically what's happening is they are allowing for an overexpression of genes. Methylation turns genes off. So because of this overexpression of genes, DNA methylation doesn't occur, and you can have an abundance of things that occur that you don't want to have. So here's how it might look like on a timeline if you develop lupus. So you have genetics, you have that predisposition, right, elevated risk, and then somewhere during your life you get exposed to a lot of things, a lot of what they call triggering events. Infections, metals, right, maybe adverse childhood experience, maybe other toxins and so forth. And so then over time you have a loss of tolerance, right, loss of immune tolerance. And then you develop antibodies. That can show up probably 9-10 years before a full-blown diagnosis. And so this keeps going on, and then you get full-blown autoimmunity, right? Full-blown lupus. And the thing that keeps it going are the mediators of inflammation. So things like all the cytokines and maybe even just like epigenetic changes that occur throughout your life. So then you have full-blown symptoms, and you might feel a worsening of symptoms until you figure out what you can do to suppress the inflammation. to stop the cytokines from doing their damage, and that's where biologics come from, and that's why you take immunosuppressants and things like that. Modern medicine has to offer there, which means they're effective, but at the same time, we really need to figure out what this is so that this can stop. This is really what it comes down to. So anyway, what I can say is that the development of lupus really hinges off of, again, a loss of immune tolerance. Antibodies are attacking you when they shouldn't, and sustained antibody production. So there are your T cells and B cells, and they've kind of gone awry, and we have poor cytokine signaling, which are the molecules that they secrete, and we have what's known as a poor defective or a defective disposal of waste material. In other words, dead cells. And I'm going to be getting into the meat and bones of that, but that's essentially what I'm going to talk about is the poor clearance of apoptotic cells and NETs, or neutrophil extracellular trapping. So we're looking closely at neutrophils, which are part of your immune system, these immune cells. They are in the spotlight of lupus because they have a huge role in organ damage and in this entire apoptosis issue that we're going to talk about here. This is like the apex of the lupus cascade. There are other things, but these are heavily involved, which is why I'm talking about it. But let's talk about apoptosis. You've heard of this term before in the health space. I know you have. It's a buzzword, but it's essentially just programmed cell death. 1 billion neutrophils undergo apoptosis every day in all of us. Okay, but in people with lupus, it's a bit different and it causes a problem. I'll tell you why in a bit. So UV light infections, toxins, endotoxins can actually increase apoptosis. Now, when this happens, essentially there are parts of the nucleus and other cellular material that get out in the open, and they're generally not accessible to the immune system, right? Which is why it's not an issue for most people. They're generally cleared out pretty rapidly. But when the burden of apoptotic cells exceeds the rate at which they can be cleared by these immune cells called your dendritic cells, you've got a problem. Instead, it induces an immune response, right? We also think that there is an issue with the production of an enzyme called DNase 1, which is also involved with breaking down this DNA of these neutrophils. Okay, so here's how this looks. I know it looks confusing and like, what the hell did you do? But like, let me break it down and I'm sure you'll understand in a bit, hopefully. So we talked about neutrophils going through apoptosis, right? Neutrophils do something else besides that, right? They release NETs, right? This net, okay? So when they're activated by infections and fungus and parasites and free radicals and endotoxins, this thing can happen where, so if we're looking at a neutrophil, this is like the neutrophil that we're talking about here, and essentially it goes into this situation where it releases this net, this trapping, okay, where it's going programmed cell death, essentially at the same time. So when this happens, like I said before, they generate a lot of molecules, granules, the histones get processed, right, as part of the DNA package, and the cell disintegrates and ruptures and releases all sorts of proteins and nucleic acids and antigens and enzymes and cytokines that are in this net, okay? And one of them is interferon, type 1 interferon, okay? So this net here is essentially what we're talking about. It's like a Venus flytrap, because what it does is it binds to pathogens that are out there, traps them, and prevents them from disseminating from the initial infection site. It also engulfs things that stimulate it and penetrate it, like we talked about. However, making too many of these nets, or not disposing of them rapidly at the right time in the right place, is pathogenic. In lupus, that's one of the issues. So I want you to direct your attention towards these plasmastoid dendritic cells. They are like the center of attention. When this net stuff is going on and they're not cleared appropriately, these cells take them in, okay, and they start to secrete interferon. So neutrophils secrete it, and then plasmastoid cells secrete it as well, and it becomes one vicious cycle. That's an issue, right? Now, these nets also stimulate these immunogenic dendritic cells, which are just a different type of dendritic cell under the umbrella, okay? And there's an increased production of these cytokines, these pro-inflammatory cytokines, interleukin 6, 12, and 23. And essentially what they're doing after these cells process these nets, they're making a package, right? They're combining with this MHC2 protein that's coming from the genetic side of it. That's what I was saying before about the major histocompatibility complex. That gene is coding for some proteins that this immunogenic dendritic cell will make, and basically it's going to package that protein with whatever it soaks up, like this net right here, and it presents it on the surface of the cell membrane, and then that activates your T cells. Okay, it presents it to the T cells. It can also present it to your B cells, which are responsible for making antibodies, essentially. However, we have these tolerogenic dendritic cells, the last form of these dendritic cells we'll talk about. And basically, these guys are producing anti-inflammatory cytokines, interleukin 10, TGF-beta. And when they're stimulated by apoptotic debris, which I'll come back to in a second. So when there's debris floating around, it stimulates these tolerogenic dendritic cells, and then they start secreting these cytokines, which are ultimately slowing down autoimmunity and disrupting the B cell and T cell proliferation and initiation of them, which you want to have, right? So they stop doing their damage. But in lupus, we know that there's a significant reduction of these cells to clear the apoptotic cells and suppress the autoimmune side of things. However, we have these tolerogenic dendritic cells, the last form of these dendritic cells we'll talk about. And basically, these guys are producing anti-inflammatory cytokines, interleukin 10, TGF-beta. And when they're stimulated by apoptotic debris, which I'll come back to in a second. So when there's debris floating around, it stimulates these tolerogenic dendritic cells, and then they start secreting these cytokines, which are ultimately slowing down autoimmunity and disrupting the B cell and T cell proliferation and initiation of them, which you want to have, right? So they stop doing their damage. But in lupus, we know that there's a significant reduction of these cells to clear the apoptotic cells and suppress the autoimmune side of things. So interleukin 6, this pro-inflammatory cytokine, is really popular when we're talking about autoimmune disease like lupus. Comes from these B cells, it stimulates your plasma cells to make a whole boatload of these green things, which are antibodies. Simple enough? So that recruits antibodies. Your interferon, this cytokine here, there's so much of this going around that it actually suppresses these tolerogenic dendritic cells, which you want to have, so that's why they don't work very well, all right? So interferon is this nasty cytokine that suppresses that. It suppresses your T regulatory cells to stop this from working, these T cells from doing their job. And it creates ultimately more of these nets that are an issue. So you'll also see that T cells make interleukin 17, which is a nasty cytokine that's highly involved with every autoimmune condition that we can think of, really. So as you can see, cells that have undergone apoptosis, they release their DNA and they get sucked up by these immunogenic dendritic cells, anaplasmastoid dendritic cells. And ultimately the cascade ensues, where you have an overabundance of T cells spitting out interleukin 17. So at the end of the day, the T cells are defective, there's an excess of B cells making antibodies, and a lot of Th17 and cytokine that corresponds with it. And we find that cytokine in the skin, the lungs, and kidneys of people who have lupus. So essentially though, this inappropriate amount of extracellular trapping that occurs here, or prolonged exposure, becomes a major source of nuclear antigens that drive the production of anti-dsDNA antibodies. That's why that antibody biomarker is so valuable. So while it can look like this genetic defect in clearing the apoptotic debris and the nets is the issue, it's only partial. You know, we have this MHC2 complex thing here, like I talked about, that has the strongest genetic susceptibility for lupus. So the issue though is mixing that with certain antigens, regardless of what it is. Nets, dead cells, viruses, metals, things like that. And that combination, that complex, is what sets up the immune system for nasty things to occur, like autoimmune disease. And we also know that there are, of course, 92 other genes that we need to consider, so it's not that cut and dried, but this is a huge part of the pie. But let's look at some of these other triggers beyond nets and dead cells that can cause this entire phenomenon. Right? We know that infections have a huge role in lupus and autoimmunity as a whole, and immunosuppressant drugs actually increase your risk for infection. That's one of the reasons why they are not sustainable long term. We know that bacteria like Mycobacterium, E. coli, Streptococcus are involved in lupus. Also, herpes zoster, which is the most common, that is what causes chickenpox, but ultimately can cause shingles later in life if it gets reactivated. Epstein-Barr virus, retrovirus, parvovirus B19, cytomegalovirus, lipopolysaccharides from gram-negative bacteria. All these can trigger, stimulate these cells right here. And from an environmental standpoint, heavy metals, of course, can do this. Mercury, silica, solvents, pesticides, we know that is involved. And, psychological stress, believe it or not, has also been linked to a 50% increased risk for developing lupus for those who have the genetic predisposition. Now, one other thing I want to mention here is UV light, okay? So not everybody has some sort of skin discoloration or rashes or lesions known as cutaneous lupus, but UV light can cause photosensitivity and cause skin issues, because what can happen is the same sort of issue here, right? It can induce poor apoptotic clearance, like we spoke about before, because you have your keratinocytes, which are cells in the skin that make the protein keratin. They release these antigens. I mentioned it before. They're called Rho and LA. Remember I talked about antibodies against those? So your skin will basically secrete that, or the keratin will secrete that. And that's how you can develop antibodies against those two proteins. But not everybody will have a positive finding for those. RNP as well, actually. Cigarette smoke, chemicals, viruses, bacterial products, all these things, like I mentioned, can interact with these receptors on these cells and your T cells and initiate this cascade. One other thing I meant to mention. So the prevalence of anxiety and psychological distress is pretty high, wouldn't you say? It's well documented in people with lupus and autoimmune conditions as a whole. And we know that emotional distress can trigger their disease flare-ups, which is not surprising. We see in Hashimoto's all the time as well. PTSD, though, of course, is something that can increase your risk of developing lupus. We actually found in a study that there was a three times increased risk of developing lupus in people who had PTSD. So that's pretty significant. So I want to wrap up here talking about kidney damage or nephritis. We can see that about 20 to 60% of people with lupus will develop kidney issues. It's pretty common. It's a multi-organ disease, but kidneys are a big target. I can tell you from my experience, I know a lot of people who have had a lot of kidney issues and kidney transplants multiple times because of unmanaged lupus. Selena Gomez is a prime example here. There's an accumulation of immune complexes that really end up in the kidney and induce this inflammatory response. That's what's essentially happening. And over time, it leads to kidney damage, of course, and loss of function. And maybe a transplant is needed. We also think that anti-dsDNA antibodies that come from this situation here can cross-react with cells in the kidney. So they attack this, and they suddenly see your kidney and attack that because of some sort of confusion. And that's how nephritis can happen there. One other thing is that you have monocytes, right? They differentiate and grow into macrophages, which are important part of this process too, even. But basically, they can recruit T cells, these guys, and dendritic cells, and they can migrate into the kidney and cause renal damage that way as well. That's why you can check for macrophage infiltration. It's a good marker to check to see if you have nephritis or kidney issues going on. because also Th17, like I mentioned before, is a marker that is highly abundant in the kidneys. All right, so that's it. That's my rant here on lupus. So I hope that was not too confusing. I know it was a lot, but I just felt like I needed to do it some respect and do the best I could with the limited amount of time that I have standing here. So thank you so much for tuning in, and I'll catch you next time.